breaking the wall between gene science and ethics. How philosophy can provide frameworks for a global biotech revolution. Francoise Baylis, Dalhousie University, Canada.
On the 9th of November 1989, I was at my first real job. It was 10 days after I graduated.
Hello, everyone. It's a pleasure to be here, and thank you for the invitation to Berlin. I want to talk to you today about an important conversation that I want all of you to be able to be part of. And in order for that to happen, what you need to understand is what my bias is. A lot of people, especially people in this audience, would be committed to the view that science literacy is important. I believe that too. But I actually think it has to go hand in hand with ethics literacy, and there's not a lot of that that's happening right now. So today we're going to have a CRISPR conversation, and the focus there is on the newest of the gene editing technologies. So, what is CRISPR? Well, first of all, you need to know it's just an acronym for a big long word, or words, I should say. And the point that's important on this side are actually the words underneath, which says precise, efficient, flexible. Or we could just say something like cheap, quick, easy to access. That's actually what's captured the imagination of scientists and others with respect to this technology, because we've been talking about modifying the genome for a very long time. But with CRISPR, it actually seems like it's a little bit closer. If we'd been having this conversation a few years ago, I might have been talking with you about other technologies, like zinc fingers or talons, etc. But today, it's CRISPR-Cas9. And you will have seen one or more of these images, depending on what it is that you typically read. Now, what I'm going to use as the framework for my talk today is the outcome of a meeting that took place in the United States in December of last year, which was billed as an international summit, specifically to look at human gene editing. Here's my conflict of interest statement. I was a member of the planning committee for that meeting. At the end of the meeting, the planning committee issued a statement, and in that statement, there were 4 conclusions. The first conclusion had to do with basic preclinical research. And basically what the committee decided is that if you're interested in doing this kind of work in cells, including sperm, eggs, embryos, the point being that you're going to do this work in the lab, go ahead, fill your boots, enjoy it. Now, that would have been welcome news to some people, because they were worried that there would be this sort of backlash, if you will, because of some recent science that had been published, and that they would be unable to continue research that they looked at and understood simply in terms of knowledge production. And it's really important, they would argue, that we be allowed to continue this work. And in that context, the committee said yes. If it's staying in the lab, go ahead, do that work. Now, this would still be controversial to some because it included doing work in the human embryo. The second conclusion had to do with clinical use in somatic cells, and somatic cells are just our body cells. So, for example, take your muscular tissue, brain tissue, etc. And the idea there was that there were a number of clinicians who had the idea that they could help individual patients. For example, a patient with Huntington's disease, a patient with cystic fibrosis, perhaps certain types of muscular dystrophy. And they're basically saying, look, we're on the cusp of being able to offer specific interventions that would offer treatments to these people so that we would improve their quality of life. It's kind of hard to be against offering treatments to people. In that context, not surprisingly, the committee says, go ahead. Now, one of the things that's important for you to understand is that a number of the scientists going into this meeting were particularly concerned that what was going on in terms of manipulation of germ cells was going to end up with a backlash in terms of the science around somatic or body cells. As I said, the committee said, if you're looking to do clinical trials, in other words, to move from your research in the lab, in the animal model, into the human, we know how to do that. We have a number of structures in place, whether you're thinking about your research ethics committee, whether you're thinking about your regulatory mechanisms, whether you're thinking about government laws, etc. And it said, we have confidence in those mechanisms. We may need to improve them as we learn more, but we know how to do that. Go ahead. The committee then moves to the third and most contentious topic going into this meeting, which is that same kind of research in the human, but in the germ cells, so in your sperm, in the egg, in the embryo. And the motivation there is, well, why would we help a patient with Huntington's and then allow them to reproduce and have children with Huntington's, and we just have to do this over and over and over again? Surely, if we're going to fix it, let's fix it once and for all. But in that context, you can also imagine that some people are saying, well, wait a second. After you've done that, what are you going to do next? And what if you made a mistake? And what if that mistake carries on through multiple generations? And what happens when you combine this technology with other technology, whether it be genetic technology in terms of gene drive, whether it be that these offspring now get, artificial intelligence, et cetera? And many people are talking about the convergence of the science and now starting to wonder, what would it mean to take over the evolutionary story? And ultimately, that's what this is about, right? If you're going to start that kind of manipulation. The committee comes up with a conclusion here that says it would be irresponsible to do that science. The fourth conclusion and final conclusion is this, which basically says, this is the beginning, not the end. We haven't had a meeting so that we can tell you that we've solved it all. Let's start a conversation, because things can change. Things can change in terms of facts, values, norms, social expectations, et cetera. And it should all be understood to be up for conversation. Now, what I want to do for the rest of my talk is take you back, and I only want to focus on what was, in fact, the third conclusion, and I've put up there the actual wording, because here's where you see your philosophical framework, if you will. And what's great about this is it's simple. There's only two things: safety and efficacy and broad societal consensus. The great thing about that is not only that it's only two elements for your framework, is that we haven't got a clue what either of them mean. Right? Now remember, I'm a signatory to this, and I think that's great. Why? Because that buys me and a lot of other people time. Time to do what? Time to think. Time to embrace the notion of slow science, especially in a context where this science has been moving so rapidly. I could put up graphs here showing you just what's happened in terms of exponential publications. I could talk to you about the millions and millions and millions of dollars invested, not only through government-funded, but private institutions, because of the prospect of this science for doing good. So this is the framework. And now I'm going to try and focus in on one thing. What does broad societal consensus mean? So I'm going to leave for today what it might mean to talk about safety and efficacy in terms of this science. Well, let me take you back to the science. Science literacy and ethics literacy. And the first thing is that we need to help people understand the basics of what's involved. And the language this time around has been gene editing. We talked about it before as gene modification, genetic engineering, et cetera. Now it's gene editing. And the reason is people think it's easier for the general public to understand things if you relate it to the computer. You cut, you paste, you move. A lot of problems with this metaphor, because that implies a level of precision that we don't quite have if we were thinking about all of the safety issues in terms of off-target effects or on-target effects, but with unintended consequences, et cetera. But this is as much as you need to know for what I now want to tell you. Broad societal consensus. How are we going to possibly achieve that? I guarantee you that if we were to have a vote here today about it, we wouldn't have consensus. If you came back and I lectured all day at you tomorrow, we wouldn't have consensus. And why? Because we come from different places, we have different histories, different ways of belonging and understanding and reading our world, different cultures, different religious beliefs, etc. So what are we going to do if we need to move towards broad societal consensus, if we imagine that that's actually a criterion for going forward with the science? Well, can it possibly mean unanimity? Absolutely not. We don't even get unanimity in politics. How would we ever imagine getting unanimity in ethics? We could only do that, in fact, if we were to actively repress minorities. What about something along the lines of probable certitude? People have advanced that as an idea, where basically what you're looking at is sage counsel, excellent arguments from wise persons. Many people think that sort of demonstrates a fair bit of hubris to think that that's how you'd go about solving the problem. And people have said something like, maybe the most we could hope for is something like a provisional commitment. Other people talk about deliberative acceptance. And there you have a commitment to both substance and process. How are we going to have this conversation? How do we imagine moving towards something like a consensus, which isn't going to be about unanimity? In that context, a number of principles come to bear. How do you have that conversation? You've got to have it responsibly. And what does that mean? That means if you come to the table to talk, you don't just sit there. And then when you don't quite like when the conversation's going, then you've got all kinds of objections. That doesn't work that way. If you're coming to participate, you participate responsibly. Self-discipline, what does that mean? It means you actually pay attention to when you're raising a concern or objection, and that it's actually grounded in some kind of principle or commitment. You're not just objecting for the sake of objecting. This isn't a game that we're playing. Respect. You want people to respect you in the context of that kind of conversation. You have to offer up the same kind of respect. Cooperation, what does that mean? It means you don't start off by looking about all the things you don't agree on. Can you find anything that you've got in common? And how do you then find that and start to lay the foundation? And then the last thing is to recognize it's going to be tough. It's a struggle. You're not going into a conversation thinking this is about proselytizing. This is about changing everybody else, because actually I've already got it all figured out and we'll just show them how to do that. So ultimately, what am I talking about when I'm thinking about the wall? I'm thinking about walls that we have in the real world. We have walls around our labs, around the places in which we do science, and we think, or some of us think, that the decision-making ought to happen within that framework because it's the scientific elites who understand the science who are going to be able to help us make those decisions. Other people think we go into a different place. We go into the boardroom. Imagine the companies that are lining themselves up to make profits off of this science. Or think about places of government and other seats of decision-making. And what I want you to think about how these spaces are gendered, how these spaces, in fact, have rules of conduct, expectations, and they are not welcoming unless you already have a whole lot of other credentials behind you. But one thing I want to tell you about the human genome belongs to all of us. So why would we think it stays inside these walls? So I want to take it out of the lab, and I want to take it out of the boardroom, and I want to move it into the kitchen. Why? Because we all spend time in the kitchen at some point, right? And the kitchen typically is a welcoming place. I come from Nova Scotia, and if you invite someone to your home, they're always in the kitchen. You can't get them to the living room, right? It's a kitchen party. The great thing about a kitchen is lots of kitchens have windows onto the world. And so it's a way of bringing people into your home, into a communal space, and think about the rules of engagement in the kitchen. They're very different from what happens as a conversation in a boardroom, in a lab. And yet, the people in boardrooms go into kitchen, the people in labs go into kitchens. We all end up in kitchens. And the great thing about kitchens is that they differ in terms of cultural diversity. This is a local artist in Canada. This is, despite colonization, a picture of what it means to have a kitchen and a particular view out onto the world. This is a kitchen in Lesotho. It's a different kind of place, but we do the same kinds of things, and we have important conversations in kitchens. Another kitchen. And maybe this is the best kitchen of all. It's a picnic. What would happen if we took down all the walls, if we had ways of making welcoming spaces where everybody gets to contribute? What kind of a world would that look like? Let me end with a couple of points. People in my area do a lot of surveys to try to find out what people think, and they ask questions in ways that are supposed to be able to help everybody participate in the conversation. And this was a study which asked about changing the genes of unborn babies and who should decide. Now, the interesting thing is that the choice given to people was actually category 1 and 3. So they were asking, should we let the scientists decide or should we let the politicians decide? Now, why they only had 2 choices, God only knows. And why do I say that? Because there actually were enough people that pushed back and said, well, I actually don't think it belongs to either of them. So first of all, we ought to think about the preponderance given to the scientific community, but we also ought to think about why was that it? The other thing to think about is they actually had opinions of their own. Why didn't they have confidence in their own opinions? Why did they think they had to defer to either the scientific community, the political community, etc.? So imagine a different kind of building. Imagine a building which has the lab, the kitchen, the boardroom. Might it look like this? Or could it look like this? On the first floor, we don't know how to talk to each other. On the second floor, we're starting to break down some of those walls. But look up above, we're talking to each other, and we're actually expanding the space out into the world. We're welcoming others. That's what you want. And this is what we have to avoid at all cost. This is what happens in architecture school if you don't know how to build a building with a good foundation. Your professor will say to you, and where are the sky hooks for your building? Buildings need solid foundations, and buildings need walls, and yet within them we can do creative things. We can take down the walls on the inside, and we might even one day think about taking down the walls on the outside. Welcome to my picnic. Thank you very much.